All right, well, we should probably get started. It's my pleasure this afternoon to get to introduce Dr. Jake Hughey. He has been with us for a few years now. He joined the Vanderbilt 3 years ago now, I believe, 2016. He said he arrived in the Department of Medical Bioinformatics. Yes, Biomedical Informatics. Always don't know where to put the bio on that, but. And I've had the pleasure of hearing him speak a number of times on time-based information, in particular circadian rhythms. And I really enjoy hearing him speak, so I'm glad I have an opportunity to introduce him today.
Oh, really? Sorry. I had my introduction ready. What did you say?
Go ahead. You can pile on more, please.
You probably said
I didn't have enough.
No, the only thing I wanted to
Oh, there's no way he said everything.
Exactly.
Yeah. The only thing I wanted to say, did you say that you were in a two-view laboratory,
Yeah,
right?
yeah.
Did you say that?
I did not.
See?
That's right. See?
So this guy at two-view is now head of the Computational Institute at Stanford.
You see that?
UCSF. Okay, and also is one, if you do a search for the top 100 gurus in machine learning or that you should follow, he's one of them. Yeah, he's not number 100. I think he's in the top 20. So anyway, Jake, we're fortunate to have you.
Yeah.
I actually did VMC.
Yeah, that's true.
Okay.
That's why no one knows how to say the department name. It's because we're in VUMC instead of VU. Yeah, right. Yeah, DVMI.
Because we had Sarah at the fellowship there, so I had to know what to say.
Okay.
But thanks for coming.
Yeah, happy to. Yeah, so Darren mentioned I do a lot of stuff on circadian rhythms, but I'm going to do something a little, maybe a little unusual, and not talk about circadian rhythms at all today, but instead talk about one thing that I really don't do anymore, about a type of research I really don't do anymore, and another type of research that we're just starting to get into, but that I think is really interesting. And I tried to make the title as vague as possible in case I didn't have time to make new slides and needed to fall back on some old ones. But I did, so we'll see how it goes. But before I get into the actual research, I just wanted to give this kind of anecdote that kind of, I don't know, exemplifies the way that stuff I like to do. And it's from this article in the New York Times several years ago by Errol Morris, who's this documentary filmmaker, and he got obsessed with these two photos. And they were taken shortly after the Crimean War. And you can see in the one on the left, there are cannonballs all kind of pushed along the side of the road. And in the photo on the right, the cannonballs are actually in the road. So this is taken by I don't remember the name of the photographer, but for a long time, only the one on the right, only the photo on the right was actually known. And then the one on the left was discovered. And the question was, which of these came first? Did the photographer come upon this road and move the cannonballs into the road, or move them off the road? And there was no evidence one way or another about what actually happened. The photographer in his notes never said. And so Errol Morris got obsessed with this question. How can you, from the photos themselves, try to reconstruct what happened? And I don't want to spoil the punchline, but it's a rather long article, but it's really interesting. And in the end, he tried so many different ways, and in the end, I think they came up with a plausible solution, a plausible explanation for which one came first and why. But I really like this example because these photographs are the data, right? And the data are all you have, and they're imperfect, they're biased, they're noisy, but you have to use the data to try to learn about the world and the system that actually produced these data. And that's what I think we're trying to do, and a lot of what I really enjoy in this place. So I have two stories that are tenuously related. And the first is from actually my PhD, which was several years ago now, but there was a paper that I was kind of spearheading in the last year of my PhD, and the paper itself only recently was published. So I really want to talk about this because I figured it would be the perfect audience for it. And then the second story is about scientific publishing and how we disclose our research. So let's talk about the first one. So the question is, the general sort of question is, how do cells create signaling dynamics that are specific to the stimulus and the concentrations? So there are a lot of pathways inside the cell, and a lot of the pathways are sort of stimulated by the same things, right? So cells, how do cells know which particular cytokine they were stimulated with if in the end you have these same transcription factors that get activated, right? And how do they know, or to what extent do they care about whether the particular cytokine, let's say, that activated them was just, present at a little bit at a lower concentration or a higher concentration? And so the way we studied that in my PhD was the particular system we studied was the innate immune response. And I'm gonna use this cartoon a lot throughout these particular slides. So a lot of what we studied was this transcription factor NF-kB, that drives expression of a lot of genes. And upstream of NF-kB, you have, for the purposes of this talk, two main ways that NF-kB can get activated, and that is through the TNF receptor, which binds to TNF, tumor necrosis factor, or you have this other group of toll-like receptors, or the IL-1 receptor. They can get activated by IL-1 beta, and TLR4 in particular, it recognizes lipopolysaccharide, LPS, and in general these TLRs go through this particular set of proteins, and the TNF receptor goes through this particular set of proteins, but they all pretty much converge on this kinase TAC1, and then they diverge again to MAP kinases and this NF-kB. And so the way, so our sort of bread and butter for studying this system was through live cell imaging. So we had cells that were expressing a fusion protein of, in this case, NF-kB with a fluorescent protein, and then we could stimulate our cells with whatever we wanted and then actually image them under the microscope and see what NF-kB did. So in this case, let's make, it worked in Google Slides. Okay, this is weird. Can I, yeah, here we go. All right, this is the only video, so it's okay if it's kind of janky. All right. So this is a cell that was stimulated with 10 nanograms per mil TNF, and what you're seeing is NF-kB move in and out of the nucleus. And this is what it does when it's activated. And it oscillates like this because among the many genes that it activates are a couple inhibitors. And so there's this negative feedback loop, and under the right conditions you can get these sustained oscillations. And with TNF you can get, these oscillations can go on, as long as we can image the cells, basically. And so let's go back to slides. So we've done a lot with TNF, and let's move on. The question was, all right, well, what happens with other stimuli, right? So we'd kind of like beaten this part of the pathway to death and looked at how it responds as a function of concentration. And in general, as you increase the TNF concentration, so here I'm showing some plots. So on the x-axis is time, and each curve here is a cell. And these are just some representative traces of cells and their nuclear NF-kB over time. And so you can see that, let's see, I don't have a, I didn't have a laser pointer. You can see that as the TNF concentration increases, you get more long-term NF-kB signaling. And in fact, many of these cells, these are oscillating like in the video I showed before, right? But what we noticed was this really striking difference with LPS and I1 beta, right? Which again are going through this other arm of the pathway, which is that at high concentrations, you get basically one peak. So NF-kB goes into the nucleus once and it comes back out and then it's done. And it does basically nothing else, even though the stimulus is still in the media. We haven't changed the media at all, right? The LPS or the I1 beta, they're still in there. And so, whereas at low concentrations, right, you see some of these oscillations, you see some of this stuff going on. And so, the way I kind of thought of it was to imagine you have a gong, right? And you tap the gong and it kind of, I don't know what the verb is for the noise a gong makes, but it makes a sound. And you know, with TNF, right, you hit it harder and it keeps making this sound longer and longer, right? Whereas with I1 beta, it was like you tap the gong and it, you know, gongs for a while. But if you tap it really hard, if you like slam the gong, it makes one noise and then it stops and just turns itself off. So that's what seemed to be happening. And that was really confusing. And so basically the rest of what I'm going to talk about is what is actually going on with this thing. Does this problem make sense? Does the reason this is confusing make sense?
And the scales on those, they're all scaled between 0 and 1. But
Yeah,
they're
right.
comparable between, so
Right.
like 1
Yeah.
the bottom
Yeah.
plot's the same one.
Yeah. They're scaled right, so each cell has a max of 1. But these, I'm only showing you cells here that are that responded to the stimulus.
Sure.
Yeah.
Is that max the same at all of the different fractions of ligand? No.
No, it varies a little bit, and it would increase kind of as you go down, as you increase the concentration. But even the ones at the bottom there that are responding to a low concentration, there it's pretty clear. It's not like 1,000-fold lower response. You know, it's a few times lower maybe.
So in the stimuli, they are specific to each of these receptors. So you cannot, like basically you have these two branches there and you have the different receptor. And so if you assume that if you have TNF alpha, that you only go through the blue branch.
Yeah.
And if you have LPS, you only go through the green one.
That's the assumption, yeah. So the question was really, where is the response actually getting turned off, as seems to be the case, and if so, where is it getting turned off and why? So we thought about how to try to figure this out for a while, and what we came up with was not new, but was new to this particular system. And the idea is just hit the system multiple times, right? So we call that primary and secondary stimulations. And so imagine you stimulate the cells once, and now in the previous experiments we just left the molecule in the media, right? But now we're only going to stimulate the cells for, you know, maybe 30 minutes. Right? Let's say we give the cells I1 beta for 30 minutes, then we change the media as thoroughly as we can to make sure it's no longer there, wait some amount of time, let's say 3 hours, and then stimulate them again and see, can they actually respond to the particular stimulus that we give the second time. The hypothesis is that if these cells that have stopped responding to I1 beta, let's say, to high concentrations of I1 beta, if they've actually turned off this pathway, then presumably they're not going to be able to respond the second time. And that's what's kind of schematically illustrated here. Imagine each cell here, we're gonna look at its nuclear NF-kB as a proxy for whether the pathway gets activated. And if you have some, what we're going to call tolerant cells, then they're not going to respond the second time. Cells that are non-tolerant, right, that are able to respond, you're going to see that NF-kB. And then just for the sake of visualization, we're just going to show these as a heat map where every row here is a cell, and the color indicates the level of nuclear NF-kB. So the direct translation here is these cells have activated nuclear NF-kB again, so they're not tolerant, and cells that are tolerant don't respond to with NF-kB. And then we can just do the whole combinatorial experiment, right, where we stimulate cells first with TNF, low IL-1 beta, high IL-1 beta, or LPS, or whatever we want, right? Then wash it out, wait 3 hours, stimulate them again with whatever we want, right? So, for example, and here I'm showing you the heat maps, right, where every row is a cell here, and the columns are time, and I'm showing you only the secondary response, only the response to the secondary stimulation. Okay, so we're ignoring what happened in the first, 3 hours or so. And so, for example, here, if you stimulate cells first with TNF, then you wait 3 hours and hit them again with TNF. This is driving me crazy. Sorry, I brought a laser pointer and I just neglected to plug it in. If you stimulate them with TNF, then again with TNF, they respond pretty much identically to the way they did the first time, right? So TNF does not induce this sort of behavior. Let's see if this works. Yes. Right, so TNF, they're just fine. Now imagine you hit them first with with IL-1 beta, high concentration of IL-1 beta. Then you hit them again with IL-1 beta. They don't respond, right? This is exactly what we hypothesized. They don't respond. And in fact, if you hit them first with LPS and then with LPS, same thing, they don't respond. If you hit them with LPS and IL-1 beta, they don't respond. Hit them with IL-1 beta and LPS, they don't respond, right? So on the other hand, if you hit them first with IL-1 beta or LPS, they can still respond through TNF, right? So you hit them here, and this pathway is somehow getting shut down somewhere. But this one is still working. And in fact, this one never shuts down on this time scale. On the other hand, right, what we saw before, if you hit them with a low concentration of IL-1 beta, then they're not tolerant, right? They're still able to respond to LPS and IL-1 beta, consistent with the initial observations. All right, so what we tentatively conclude from this is that there's something inside this blue shape here that is inducing this sort of behavior, right? We've using these stimuli and their responses, we've mostly narrowed it down. It has to be something common to the stimuli that go through these receptors, right?
But at which concentrations are these done?
These are at the high concentrations. Yeah, so the low concentrations don't induce this behavior, but something at the high concentration, something in here at the high concentrations of the stimuli seems to be doing it.
Do you have a way to actually measure the occupancy of this receptor? So could you use a fluorescently labeled IL-1 or
Yeah.
to actually see, are they actually completely saturated, the cells?
Maybe. We didn't do that, and I don't know how technically feasible it would have been, but we'll get close. We'll see what we think of where we end up going. All right, so now let's see, how much further can we narrow it down? All right, so for that, and this is the part really that I had almost nothing to do with, and my co-authors did an awesome job with. So we took advantage of optogenetics, and in particular, there's this Cry2 oligomer, this piece of Cry2, which aggregates, which basically oligomerizes in response to light. And again, we weren't the first ones to use this, but we kind of co-opted it for our system by making fusion proteins to either MID88, so this is this protein up here that is kind of binds with the receptors, or to TRAF6, which is downstream of IRAK1 and basically right upstream of TAC1. And what happens is, so the mechanisms by which these particular proteins, MID88 and TRAF6, activate the pathway is not totally clear. But what ended up working was that we could fuse this Cry2 oligomer, this Cry2 to MID88 or TRAF6, and then shine light on the cells, and that induced aggregation of either MID88 or TRAF6, and that could activate the pathway. And so again, what we're trying to do is narrow down where this behavior is actually happening. So again, you do the same primary and secondary stimulations, except now instead of hitting the cells with some cytokine or some molecule, you just shine light on them. So in this case, they're expressing opto-MID88, and what you can see is that if you stimulate them with I1 beta, and then again with I1 beta, they don't respond, right? So that's what we've already seen. If you stimulate them with I1 beta, and then then light, let's see, I1 beta and then light, so you're activating MID88, they don't respond, right? On the other hand, if you stimulate them first with light through MID88, and then again with light, they still don't respond. Okay, so something downstream of MID88 is inducing this behavior. On the other hand, what about TRAF6? Okay, now we're going further down. All right, now you can see a difference. We stimulate them with I1 beta the first time. If you stimulate them later, the second time with light, they can respond. So NF-kB shows a strong response. Right, so you've stimulated them with I1 beta. So you know they can't respond to I1 beta, and yet they can respond through TRAF6. Right? So the conclusion is that we've basically squeezed this a little bit more. So now you imagine you stimulate them first with I1 beta. The pathway essentially is unresponsive to further I1 beta. But you can bypass the behavior if you activate TRAF6. So it has to be upstream of TRAF6 somehow and downstream of MID88. And there's really at that point only two things left. And that was IRAK4 and IRAK1. And these are both kinases. And in fact, what I'm not going to show you, but that we verified, was that IRAK4 still became active, still became phosphorylated after a secondary stimulation. So even if the cells couldn't activate NF-kB the second time, they were still activating IRAK4. On the other hand, IRAK1 basically got degraded even after a primary stimulation. So it didn't completely disappear, but it got degraded significantly, and this absence of IRAK1 basically correlated with an inability of the cells to respond the second time. So the first time you hit the cells, IRAK1 would go away. If you stimulated them with a high concentration of I1 beta, IRAK1 would go away, and then when you stimulate them the second time, there's no IRAK1 there. And NF-kB didn't do anything. So at least we have a correlation that, okay, maybe you need IRAK1 in order for this pathway to get activated. Does this question make sense? So the next experiment is relatively clear. We want to try to rescue it. We want to induce IRAK1. And so what I'm going to show you are the same heat maps with IRAK1, where the cells are expressing IRAK1 under an inducible promoter. Now the trick here is that we want to induce IRAK1 after the primary stimulation. So here's a control where there's no doxycycline. And now I actually am showing you the full primary and secondary response, again, where each row is a cell. So we hit the cells with IL-1 beta the first time. They all respond pretty strongly. And the second time, not much happens. Okay, again, consistent with everything we've seen before. And just another way you can visualize this is here is another type of heat map, I guess, a 2D density plot where the x-axis is the the amplitude of that first peak to the primary stimulation, and the y-axis is the amplitude of the response to the second stimulation. So what this is showing, that all the density is down here because the cells are not responding. The secondary response is very low. All right, so now we can do 2 more experiments. All right, the first is where we add doxycycline right at the time of the primary stimulation. So what we're trying to do is make sure that the cells do whatever they're going to do in response to IL-1 beta, and then they start making new IRAK1, such that IRAK1 there's some new IRAK1 around by the time the secondary stimulation comes around. And in that case, you can see actually they respond. So first of all, they're doing a lot more stuff here, right, when IRAK1's getting induced, but also when you add IL-1 beta, and again you can see it here as well, they're not tolerant, as in NF-kB is now responding to the second stimulation. On the other hand, if you add doxycycline 24 hours before you do this, right, you add doxycycline way back here so that you now have lots of IRAK1 hanging around at the time of the first stimulation. Okay, now whatever happens to that IRAK1, it happens to the induced IRAK1 too, because they're not able to respond. And you can see it here as well. You have to induce the new IRAK1 after that primary stimulation in order for it to have any effect. All right, so we can go a little bit further and actually ask, this is not exactly what you asked, Gregor, but it's as close as we got, I would say, to see actually what happens with IRAK1. And in fact, when you express a fusion GFP, or maybe it's Clover, to IRAK1 in any case, you see that when you stimulate cells with IL-1 beta, IRAK1 actually does this sort of aggregation, clustering. So there was no optogenetics here. This is actually what the protein seemed to be doing. And in fact, if you look at, so you can quantify this sort of clustering, sort of how speckled IRAK1 becomes. And you can see that, so here I'm showing you the NF-kB response again, and here I'm showing you the quantification of IRAK1 clustering. And just sort of, if you discretize this into was IRAK1 clustering low or high, you can see that actually the cells in which IRAK1 doesn't cluster as much are the cells that an NF-kB response seems to go on longer, where there seem to be more peaks, right? So this is implying that it's this something related to this clustering of IRAK1 is at least correlated with shutting off the response. Yeah, Ken.
I'm just wondering, for all these reported experiments, the reporters put into the endogenous locus?
Yeah, so for P65, so for NF-kB, it's not in the actually, I think none of them are in the endogenous locus. The cells are, in this case, these cells that we're showing are basically the only functioning subunit they have of NF-kB is the one that's the fusion protein. And that is true, I believe, also for IRAK1. We added IRAK1, or what you're going to see later, different versions of IRAK1 to the IRAK1 knockouts. But they're not in the endogenous locus. It's from lentivirus.
Are you surprised that, given the different expression levels, that the regulation sort of recapitulate the native?
Yeah, so, well, with IRAK1, I mean, I have very little intuition about IRAK1, but for NF-kB, it's definitely true that the baseline level of NF-kB has a big effect on the dynamics of the signaling pathway. So if you add, and in fact, that was one of the reasons it was, for a long time, it was actually hard to get the system to behave the way we think the native one behaves, is because the NF-kB is being expressed under its endogenous promoter and is pretty low, and is therefore pretty low and more difficult to image. IRAK1, it's hard to know. How, I guess we have some Westerns. The inducible IRAK1 is definitely being expressed at a higher level than its native protein, but it's hard to know.
I mean, the only reason I ask is because there's a protein regulation, and
Yeah.
there's also a transcriptional regulation, right?
Yeah,
And
Yeah.
I don't know how those
right,
given that exogenous factor.
right. Yeah, it's true. All right, so we have this clustering, and at the same time, we know IRAK1 is a kinase. And so, again, the question was, well, to what extent is the actual kinase activity of IRAK1 required for this sort of behavior? And so for that, we could generate a kinase-dead version of IRAK1. And again, I'm showing you just the heat maps of the response to the secondary stimulation. And you can see from both these types of plots, if IRAK1 has no kinase activity, then it impairs tolerance, as in the cells can now respond more strongly to the secondary stimulation, right? Which implies that this kinase activity is not totally required, but at least important for turning off the pathway, right? And in fact, this is sort of quantification, right? The kinase-dead, so here's images of cells stimulated with I1 beta, right? And you can see here's the wild type version of IRAK1. It does this clustering, this speckling. On the other hand, the kinase-dead version doesn't cluster. It doesn't go through this sort of speckling, right? So what we interpret from that is that this version of kinase-dead IRAK1 can't induce tolerance to the same extent, and it can't cluster. So again, we're not totally sure what this what is exactly happening in this clustering and how it relates to the actual receptor. It's possible that all these proteins are kind of on the membrane itself. But at least we can now come back to that original observation, and instead of IRAK1 wild type, we can add the kinase-dead IRAK1, and now, right, it's not perfect, right? The cells still aren't oscillating forever the way they do with TNF, but at least you can see a difference, right? The NF-kB activity goes on longer at higher concentrations than it did before. So it's not everything, but at least this IRAK1, and in particular the kinase activity of IRAK1, is involved in shutting off this pathway.
So when you look at the slower IL-1 stimulations, how does this clustering changes?
Yeah.
I mean, is there like a kind of dose response on the clustering, and then?
Yeah, it's a good question. Now I'm not sure. I'm sure we did. The Royal We did that experiment. And now you're exposing the fact that I did this 4 years ago and didn't do the clustering. We didn't know anything about the clustering yet. So I don't want to say it for sure. That's a good question, though. So our sort of.
Is there one cluster per cell?
No, there are lots. So, yeah, and I don't think we quantified the degree of speckling, but I don't think we quantified, and this is only epifluorescence anyway, so we wouldn't be able to really quantify the number per cell probably reliably.
Those are sort of endosome, lysosome type of things, right?
Yeah, I don't know. They persist for hours, and they're still around at the time of the secondary stimulation. But we didn't really try to manipulate, to try to decluster them in any way. So it's hard to know, and I don't think we did any sort of, co-fluorescence or co-staining to see what other sorts of proteins might be in those aggregates. But at least, the sort of cartoon understanding we have of this, based on these experiments, is that at low concentrations, IRAK1, the signal goes through IRAK1, and you can get these sustained oscillations in NF-kB. Basically, the pathway is still active. Whereas at higher concentrations, something changes. Again, we don't totally understand this mechanism, but something changes such that IRAK1 basically turns itself off, as far as we can tell. And that makes the pathway refractory to any further stimulation. So hopefully that's just one example, right, of how we can use the dynamics of a pathway in live cells to understand the network structure, right? And we didn't totally come up with the structure from scratch, right? We were also very dependent on knowing, roughly the organization of, TNF-R, mid-88, TRAF, and so on, right? But sort of using what we knew before, we were able to kind of bootstrap our way to figure out where this certain behavior was coming from. And then we can use optogenetics to manipulate different points, right? The different stimuli give us one way into the pathway, and the optogenetics give us even finer control. And then IRAK1, in this case, seems to be important for both propagating and actually terminating, in certain cases, the signal. All right, any more questions about that part before I totally change topics?
Sorry, one more.
Yeah, please.
So in the beginning you say that IRAK1 is degraded, but then later you say it's clustering.
Yeah.
So when you do a fluorescent analysis of that over time, do you actually see that it's degrading, or is it just clustering?
Yeah, it's definitely getting degraded. By Western, you can see the band. Again, the quantification, I'm not totally sure, but you're definitely losing some IRAK1. Abundance of IRAK1 protein is definitely going down. So my guess would be, and I don't know, I would say maybe it goes down by 90%, or I don't know, who knows with Westerns, but
when you look at the fluorescence.
Yeah, right. So I would say, right, there's two things going on. One, you're losing abundance, right? And then second, of the stuff that's still there, it's in these clusters. And what I didn't show is from the Westerns, you can see it's getting post-translationally modified as well. And it was known but previously that there are a bunch of phosphorylation sites on IRAK1. It's probably getting phosphorylated by both IRAK4 and by itself. And so we don't know exactly which phosphorylations are necessary for it to sustain activity and do its different functions. But that's kind of, what we think.
So I'm sure that you thought of this way of regulating the network signal as, so we have IRAK1 acts as a circuit breaker, like a fuse.
Yeah, that's a way to think of it.
Are there any other examples of signaling?
Yeah, it's a good question. I don't know of any. I mean, there are certainly, ways that sort of autoregulatory and negative feedback loops. And the behavior, we're sort of calling this tolerance. We're sort of leaning on a lot of literature on this phenomenon of endotoxin tolerance, which has a bunch of mechanisms that people have proposed for it, none of which were this. And this is only right in our particular cell type and cells, so who knows what's actually going on. But I don't know of any exact examples where you have one protein that seems to be almost like as far as we can tell, almost like by itself regulating the flow through its kind of arm.
So endotoxin tolerance, this is a septic shock, right?
It's related to septic shock, where it's a similar sort of idea in principle, except with instead of stimulating cells, you stimulate maybe mice or something, where you stimulate mice with, say, a low concentration or something of LPS, some amount of LPS is not going to kill them. And then the second time you stimulate them with LPS, they don't die. So whereas like it's sort of the first time you either stimulate them with nothing or with a little LPS, and the second time you stimulate them with a lot of LPS. And if they first got a little bit of LPS, then they're going to survive a lot of LPS. And that was a sort of idea of endotoxin tolerance, that something in the pathway kind of turns itself off such that it's relatively insensitive to further stimulation.
Maybe one of, I don't remember much about this, but someone mistaking septic shocks kills about 40,000 people in the States right now.
That could be. I don't know the numbers. Yeah. Yeah. Right. So in general, immune systems really care about turning themselves off. And when they don't, you can have autoimmunity and cytokine storms and all these other things, right? So it really matters how these things do what they're supposed to do, respond to an infection, and then stop before they cause real damage to the tissues.
Did I miss it? When you
Probably not.
introduced the kinase-dead form of IRAK1, it did not inhibit the primary response to IL-1 beta. Is that
That's
correct?
right. Yeah.
So how do you, most of the time I see with kinase-dead, they act as dominant negative
Yeah.
kinases. So what do you think of that?
I mean, the interpretation is that the kinase activity of IRAK1 is not actually necessary for activating signaling through the TLR and IL-1R pathway.
But is IRAK1 itself necessary?
Yeah.
Can you knock out
Yeah.
IRAK1 and
Yeah.
then lose it?
Yeah.
And you don't get any? Interesting.
Yeah.
So maybe scaffold type?
Yeah, and again, there's so many things these proteins do. People have really latched onto NF-kB and the MAP kinases because they're so easy to study. Stuff that happens further upstream, just a little bit downstream of the receptor, that is so hard. And so there's so many phosphorylation sites and other things that could be going on. Just post hoc hand waving, right? You can see why. In the total absence of IRAK1, you cannot activate this pathway. But if you have IRAK1 that can't, or that doesn't have kinase activity, you can activate the pathway. It's just a little bit slower to turn off. Cool. All right, then that leaves just a little bit of time for me to talk about the second one, which is after we do this research, right? So for example, research I just showed you, how do we actually disclose it? When do we decide to share it, and where do we share it? And one particular place that is starting to become more popular is bioRxiv in particular, so preprints in general, right? So the idea of a preprint, actually, can I just get a show of hands? Who has heard of preprints or bioRxiv? Okay, excellent. So then I don't have to give too much background. It's been pretty much standard in math, computer science, physics, other fields for, I don't know, 20 years maybe, and is only starting to become a thing in biology. And this is from a recent paper where they looked at basically the growth of bioRxiv. And in a number of fields, bioinformatics and neuroscience are kind of leading so far. Preprints are becoming more common, right? But the thing is, I don't know, you can see the scale here, right? This is a cumulative number of preprints. As of the end of 2018, it's in the thousands. It's maybe like total on bioRxiv, a couple thousand per month. I don't know, it's probably higher by now. That's a drop in the bucket compared to like all the peer-reviewed publications that come out each month, right? And so, okay, right, the other point is this is not just all garbage that's going on in bioRxiv. because, well, at least if it's garbage, it's also published garbage. So, you know, some 70%, let's say, on a long time scale are showing up in a peer-reviewed journal. And the, you know, half-life is maybe a year or so until from when something shows up in bioRxiv to when it gets published in some peer-reviewed journal, right? So the question that I was kind of wondering about when I was, I don't know, seeing this, and I had another experience in a journal where we really needed to go to this particular journal, but their policy was they basically don't accept anything if it's released as a preprint, and that kind of pissed me off. I'm biased, right? This is not going to be, I'm not trying to hide this, I'm biased, I like preprints. And so I thought, all right, how can we convince people or show them some evidence that preprints are like nothing to be afraid of? And so the sort of really naive question that we asked was, how does releasing the preprint relate to the outcomes as far as we can quantify them right now of the peer-reviewed article? Because I've talked to plenty of people, right, that are really afraid of getting scooped, right? You put something out there before it's been peer-reviewed, right? Someone else could try to do it and get it published before you. So that's what we're going to look at, if we can try to establish any evidence for that. And so this is an observational study, but we had about 74,000 peer-reviewed articles that we took from as many journals as we could that were on PubMed, that were published in the last, in about a 4-year period, shortly after the start of bioRxiv. About 5,400 of these articles had a preprint. They came from about 39 journals, and we were getting the data from a bunch of different places. I'm happy to go into more of the details, but I kind of just want to jump to the punchline since we don't have a ton of time left. And the metrics that we're using, right, for outcomes, right, are the Altmetric Attention Score, which is not exactly just how many tweets you had, but it's a weighted sum that factors in social media, mainstream media, and so on, and number of citations, right? If there's anything scientists really care about with their work, many of them are going to latch onto this number of citations. And so, for example, here I'm just showing you density plots, just giving you an idea of sort of the distributions of a subset of the journals. So these are, smoothed density plots of the attention score for several journals and their citations. And I'm just grouping them just crudely by year here, right? So what I want you to see is that, first of all, the x-axis, this is log transform, right? So the attention scores and the citations span orders of magnitude. And the attention scores really don't change much over time. So an article published 5 years ago doesn't necessarily have a higher score or lower score than one published last year. Whereas with citations, you see the behavior you expect, right? Older articles have more citations. So the distribution is sort of shifting to the right in older papers, right? And as you expect, some journals, the papers in those journals tend to get more citations than papers in other journals, right? So these are the outcomes we want to try to model. And again, this is an observational study, so we're going to try to shove a bunch of things into the models we can to make sure we're not getting confounded. But the primary variable we're interested in is, did this article have a preprint or not? And then we add a bunch of other things that have previously been shown to be associated with citations, with the number of citations, and of course publication date. I can go into more detail about anything if people have questions, but I think I'll just skip this. So one thing you want to know within a journal, right? Some journals cover, let's say, Nature Methods, right? They publish a bunch of different types of methods: computational methods, experimental methods, sequencing, stuff with mice, right? And it could be that if something has to do with CRISPR right now, right, it's going to get a lot of citations. And maybe only stuff related to CRISPR goes on bioRxiv. So you want to somehow control for the different subfields that articles are getting published in within a journal. And it turns out we can do that using something called mesh terms. They're on PubMed. So here is one of my favorite papers. And if you look on PubMed, if you open this window, you can see that mesh terms get assigned to almost every paper. Not immediately, but eventually. And these are added, these are terms, this is an ontology, these are terms that describe what the paper's about, and they're added manually by, I don't know where these people are that do this. I don't know if they're actually in Bethesda or if NIH hires people from, outsources it, but someone manually adds these terms and says what this paper's about. And so we can basically calculate principal components of these mesh terms to say roughly what scientific field was this paper. So here, I'm just showing you the top 2 principal components for a few journals. Each point here is a paper. And what this is saying is that there are kind of, for example, in PLOS computational biology, there are 2 main axes, sort of what papers are about. And I'm not going to go into more detail with that, but we can use these principal components basically as covariates in the model to try to make sure we're not getting confounded by this. All right, so here is one of the main results. So we fitted these linear models with all these terms, right? Trying to see how does attention score and citations depend on whether an article is released as a preprint. And here I'm showing you the number of articles we actually had for each of these 39 journals with and without a preprint, right? You can see the numbers are low, right? Again, like I was saying before, the number of preprints we have right now is kind of dwarfed by the total number of articles. But adjusting for all the other variables that we've put into the model, in most cases, right, papers that have been released as a preprint have higher attention score and higher citations than papers that don't have a preprint. And so here I'm showing you the point is the mean, and the error bars are the 95% confidence interval of the prediction for that particular, or the predicted mean for that particular metric, right? And you can try to look at to see what journal you recently published in. But right, so these are sort of absolute effect sizes, right? But we can quantify the relative effect sizes too. This is basically the coefficient from the model, and you can think of it, I don't know, if you're used to thinking about differential expression in transcriptome data, you can think of this as a fold change, right? So how many more times citations, or how many more tweets kind of do you get for your peer-reviewed article if your article was preceded by a preprint or not? And so this gray line here, this is a line of no association, and what you can see is a lot of stuff is to the right, right? So if you kind of average it, you can get to the fold change is about 1.5 for attention score, and it's about 1.37 or something for citations, right? So adjusting for when the paper was published, how many authors you had, how many references you had, all this other stuff. That's the difference.
It's hard to scan the journals really quickly.
Yeah.
Did you guys get a sense that you get more benefit for a preprint server if it's a method versus experimental result? Like I'm seeing cell elimination methods at the top.
Right. Yeah, so give me 1 second, I'll get to that as close as we got. So I can just skip over this. This is what I said before, right? About 50% higher attention score, about 37% more citations. Oh, sorry that shifted. That's a PowerPoint thing. Independent of all these other effects that are also there, right? Okay, so this is as close to your point as we've gone so far, which is to look at how does the fold change for a particular journal relate to whether that journal is open access or not, the journal's impact factor, and actually how many of those articles are released as preprints. So we haven't done anything like, is this a methods paper or is this not? That's going to be harder to do on a large scale like we have so far. You can certainly Nature Methods is an outlier, and I don't know.
The other ones at the top were like bioinformatics was at the top.
Yeah.
I don't know. It's like trying to see. If
Yeah.
maybe.
It's a good point. I haven't thought about how we would exactly quantify that. But in any case, what we see from this so far is that there seems to be no relation between these factors, the journal's impact factor, whether the journal's open access, and the difference between papers with and without a preprint on bioRxiv. Now, obviously, the caveat: this is not a randomized trial, right? This is purely observational. We don't know how many citations and how much attention these papers would have gotten if they hadn't gotten a preprint on bioRxiv, right? There are so many reasons that having a preprint just could be a marker for something, right? It's not related, as far as we can tell, to how senior the PI is, but there could be plenty of other reasons you could see this. And of course, these are just short-term effects, right? These are not anything close to scientific impact, whatever that means. But at least I would say, based on this, I don't see any evidence that people are getting scooped and that's damaging their, citations. Yeah.
Do you feel that preprints are tweeted more by the community versus published papers, which is more tweeted by the publisher?
Right. So we haven't looked at the tweets for the preprints themselves. So everything I'm showing you is actually just a peer-reviewed article. There is so it's hard. So I guess I haven't looked at that. I don't know. You could certainly imagine people who like preprints also like tweeting about stuff.
Right.
And so that could definitely explain part of the effect of attention score, right? So I put a preprint, I tweeted about my preprint. We don't necessarily care about how you tweet about your preprint, but maybe that means you're also going to tweet about your article when it comes out.
But
And
the citation also depends on popularity.
yeah, well, that's definitely true. But what I didn't show you all the data, but at least in this data set, attention and citations are not very strongly correlated with each other. They're pretty weakly correlated. The Spearman is maybe 0.18 or something. And the fold changes are also not correlated with each other, which says that that those are really different measures, and that something that could explain a behavior like that for attention probably wouldn't explain much of citations.
Was there any sense of how long it took for a preprint to become an article as having any kind of?
Right. So we did look at that, and what seems to be happening, or what we saw, is that for the attention score, actually the longer between when the preprint was released and when the article was published, the higher the attention score of the article. On the other hand, there was really no effect of citations on citations. So, an article that came out, 6 months later or a year later, the difference in citations seemed to be the same. It was more like, did it have a preprint or not?
It's surprising because I would think that the
Yeah.
journal that take the longest,
Right.
you get the most
Yeah.
citations for,
Yeah.
usually.
It's hard to know. It's hard to know we don't, again, this is totally observational. We have no mechanism. So it's hard to know what would be the reason for this, and how to rationalize the fact that it doesn't the time doesn't seem to be related, and it just seems to be a preprint. Maybe this is just going to manipulate people to put a preprint like the day before their paper comes out, right? That would be, if you were going to game it, right? Is if you don't care about tweets, if you only care about citations, and you believe this to any extent, then you're just going to put a preprint.
You cannot do that, right? Because a lot of publishers would not let you.
Ignoring the consequences, or like how editors and publishers will feel about it, just based on these results. If you're going to game it, you could try to put a preprint like right before. That's nonsensical, but I don't know. It just shows that we don't know what's going on.
So did you put these on archive?
Of course. Unfortunately, the first one I talked about, bioRxiv wasn't even around when I was finishing, or maybe it just started in grad school. So unfortunately, the first one was not on bioRxiv, but this most recent one obviously is. So with that, I just want to thank everyone who was involved in all this work. The first paper, like I mentioned, was really the last year of my PhD, and then Miley and Helen really just killed it over the last few years. And so I was just so thrilled to get them involved and to have them finish it. And then the recent stuff I talked about was done with Darwin Fu, who's a postdoc in my lab. So with that, I would love to take any more questions. And I just realized I've not been repeating the questions at all, so sorry for the people on video.
So, just a quick question follow-up.
So,
Yes, sir.
Okay, there we go.
I'm just kind of curious, so bioRxiv hasn't been around for that long, but I wonder what the effect would be on preprints and then like future careers, or like if there's a way to quantify that.
yeah.
Do you have any idea of what that might be?
Yeah, so questions about the effect of preprints on career outcomes. Yeah, it's too early to know for sure. So NIH now accepts preprints as a research product on your biosketch, I'm pretty sure, and also in grant applications. There was Michael, a postdoc in my lab, just mentioned that there was another preprint that came out that was I think trying to look at that exact thing. I think it was looking at people that were looking for academic jobs and how basically what were the correlates of like whether they got a job or whether they got an interview, how many interviews they got, whether they got a job. And I think they were looking at how many papers did they have, how many preprints did they have, and so on. I haven't looked at it myself yet, but I think it would be really interesting. There's just so many like self-fulfilling prophecies and so many biases and that sort of thing that's hard to know for sure. Yeah.
So in your signaling data, you were doing live cell imaging, so you have like dynamic data, but you started off talking about the cannonball photos, where you basically have two snapshots and
Right.
trying to deconvolve what happened first. So what was the punchline of that? How did they figure out?
Oh, I'm not going to spoil it here. You can. I don't want to spoil it. But there was. They did come up with something that is plausible and that doesn't rely on any external evidence. That is something from the laws of nature and looking at those photos that you can believe.
So what do you think is the main difference between looking at data where you do have a time course versus snapshots in time and trying to figure out things that might happen?
Well, right.
Right, 'cause I
So-
think those are two different approaches. Like the cannonball picture is one thing.
Yeah, I mean, I guess I might push back a little and say that actually is dynamics. You just don't know which one came first because it's the same exact area, right? I guess the difference is with like a lot of the times the thing we sell with live cell imaging is the fact that you can follow the exact same cell, and you know if you want to look at two reporters, you know what those two reporters are doing in this exact cell. Whereas, right, even with something that's single cell and then in principle can look at dynamics like flow or CyTOF, you only have that cell once. And if you see at one time point 50% of your cells look some way, right? And then the next time point 25% of cells look that way. You still don't know, well, which one's changed, right, and what we're doing what. Whereas with live cell imaging, you're much more limited in the bandwidth, but you're really like locked on to this cell, and you can follow this population of individuals over time. So that's kind of how I And in that sort of analogy, it's like with that valley, right? You have one valley, you're locked on, you just don't know what the order was. But I guess that's what I would say is one of the main advantages that we try to take advantage of, or that we try to use with the lifestyle imaging.
Right there, you're on.
So in the publication story, clearly could continue to be a prospective study. Are you planning on keeping that going?
I mean,
so one of the things is the citations are going to keep accumulating over time.
Yeah.
Especially because it's a relatively recent window of
Yeah.
those papers. I'd be curious to see
Right.
how they turn
Right.
out as we go on.
yeah. So the question is about how the publications are going to change over time, and right. So okay. At first, I thought you meant by perspective. I meant you want to try to run a randomized trial of preprints. Which I would love to do. I don't think it'd really be feasible, but certainly you could imagine you can look at citations over time. The difficulty with the APIs that we pulled the data from right now is that they don't provide historical data. So we only have each article's citations basically right now, regardless of when it was published, and that's why we did the analysis the way we did. Ideally, you'd want to know the full time course of all the citations. So basically, and this could be done in principle for some of the articles we have, but not all of them. You know when the articles, for a given article, when were the articles published that cite that thing? And from that, you could reconstruct the time course and say, does it matter at various time points? And 5 years from now, what is the effect, if any? So for now, we just have to wait. But you could also say, right, there are so many reasons why, again, that this sort of differences could change. In 5 years, if the culture of preprints is different in biology, then these effects could evaporate. Who knows?
Have you looked at archive data?
Not, so there was we haven't. There was a study last year of archive data on, so the question is, have we looked at archive, which is the preprint server for CS, physics, math, and so on. And there was a study last year looking at a similar thing for papers in computer science that were submitted to CS conferences. Sorry?
I didn't think people actually published.
Well, so they designed it slightly differently. So in that case, they looked at articles, they looked at things that were posted on archive either before or after getting accepted to a CS conference, which is kind of the closest analog to what we have in biology. And in that case, they saw a similar difference. That papers that were posted on archive prior to getting accepted had more citations than articles, or than things posted to archive afterwards. But there is a section of quantitative biology on archive, but we haven't pulled stuff from there yet.
So Jake, you didn't tell us why you're biased in favor of bioRxiv.
Well, in general, I have a problem with some of the abusive practices of some of the publishers, and I just like sharing stuff freely, and I don't know, just maybe naively don't think people are going to try to scoop me, so I'm not worried about that. And so I just like, I don't know, I feel like it accelerates. The peer review process is so long and painful, and putting a preprint out there is like getting a quick win.
Yeah, I can give you a review picture. I can give you a perspective. Maybe a longer stay, in science. You can put stuff out. There wasn't a bar, but you can talk about stuff for publication. You can just be secretive. You get scooped either way.
Yeah. It's true. Yeah. Yeah.
Because with bioRxiv, you can point there and say, I have done this first. It's there. There's a date
Yeah.
on it. It's been out there since then from me.
Yeah. I would also just say, like this, like the dates, there are so many things that go into scientists' perception of priority. And the date that something shows up, if we put something up, I mean, so for example, in the case of this preprint stuff, like there was another preprint actually that differed in some of the details, but that has shown a similar thing. And ours came out about the same time. It's happened numerous times that related papers have come out around the same time. Yeah, it's going to happen. Yeah.
Okay, Mark and then Oscar.
Yeah, so there is a danger. I mean, in math now these days, you see people publishing half-baked things on the archives
Right.
and claim priority.
Yeah.
So,
Right. Yeah, that's true. And to some extent, that's I would imagine, or I'd like to think some of that is honest, because depends on, does peer review work? Like, does it make the paper better? In which case, you would hope that if their peer reviewers did their jobs, then the thing that gets published should be better, more complete, more solid than the thing that gets put on a preprint. You can definitely imagine, humans like to exploit holes, and so you can certainly imagine people trying to establish priority by putting really crappy stuff on there. You would just hope that the system would kind of frown on that and say, and people would recognize, oh, that's a crappy job. And I personally, what I've noticed is I am way more anxious about putting preprints up than submitting a paper to a journal, because preprints have a DOI. They're up there forever. They're never coming down as long as the internet exists. And my name is on that thing. And so I do not want something wrong with that. Whereas I can submit to a journal, 4 people at most are going to see this, right? Who cares about that? Like there could be something wrong. We'll fix it. No one else will ever know.
So can you retract from bioRxiv?
No. You can update it. You can add a new version, but the old version will always be up
By
there.
the way, that was wrong. Never mind.
Right.
Oscar, yeah.
I was going to say, do you think that posting a preprint and then tweeting about it and having a tweet storm about it would motivate editors to upset those papers and to pursue those things?
It's a good question. I've heard anecdotes of editors seeing preprints and then asking the authors to submit their paper to their journal. It's never happened to me, so I can't vouch for how often this happens. It kind of feels like a sort of I don't know. I can't imagine it happens very commonly now, but in an ideal system, it might.
The only time it happened to me, it was from a journal that was no good for publishing it.
Okay, right.
Auto-generate
Right.
information.
Yeah.
It was spam
Yeah.
worthy.
I mean, most of the time, I guess if you have a couple journals you're considering and you want to try to test the waters and see which one bites, maybe that would make sense. But most of the time for me, by the time we put up a preprint, we know where we're submitting. And, we know our first choice, and if that doesn't work, we have a couple other possibilities. And if an editor tweeted or emailed out of the blue and said, "I'd love your preprint, please submit," okay, we'd probably listen, but I'm not counting on that happening too frequently.
All right.
Okay, thank you very much.
Thank you, Jane, for the good reason.